Right.
Same my side, yes.
Okay, Melissa, I think we're good to go now, right?
Yes, we can start.
Okay, excellent. So first of all, a warm welcome and a good afternoon to yet another AVMI webinar this afternoon. We've got a jam-packed session. First of all, I'd like to take this opportunity just to warmly welcome our AVMI membership who's managed to take time out of their very busy schedules, I'm sure, to be able to take the call this afternoon. So, yeah, I'm very excited to be able to introduce to you a colleague and a friend who will be the presenter for this afternoon's webinar. Her name is Dr. Subhadra Dravida. She's also joined by her colleague, Dr. Shri, who is the business development manager at the company. But they're here to talk to us today about a very important concept, which is really the testing of neurotoxicity and neurovirulence as early as possible during the development of a vaccine, or for that matter, a therapeutic. So just a little bit about Subhadra. She's the founding member and at the moment the CEO of Transcell Group. If you look at the resume, you'll be very impressed. She's definitely an inventor with over 15 years of technical expertise in stem cell technology, processes, as well as formulations. And obviously, over the years, this has culminated in quite a bit of peer-reviewed publications. But more importantly, she's got over 27 patents to her name in the fields of drug discovery and regenerative medicine. So I think we can all share this afternoon that we have an expert at hand that will take us very carefully through this very interesting topic. So she's here to introduce us on a novel product that they've developed at Transcell Group, which is really called NeuroSAFE. And hopefully it's the first in a series of new products that they're trying to put onto the market, following a new approach and methodology to really evaluating neurovirulence and neurotoxicity early on during your development program. So we are very honored to have Dr. Subha with us this afternoon. Just quickly before I hand over to her, just in Subha, we'll give you 40 minutes just to run through your presentation. Hopefully that's more than enough time. If you need a few more minutes, that should not be a problem. After that, we'll open up the panel and allow the audience to ask you as many questions as they would like in terms of your topic of today. And then I'd also like to encourage the membership group, maybe just to post your questions online. I could run through them towards the end of the presentation, especially if we press for time. But certainly without any further ado, Dr. Subha, over to you for your presentation.
Thank you so much, Dr. Ebrahim. It's so lovely, to again come back to your team, and then it's an opportunity for me to, share our story on NeuroSAFE tech. Yeah. Give me a second. Yes.
Subha, if you don't mind, I think your presentation is not in presentation mode yet. There we go. It's just.
I've put it on now. Yep.
Yeah, perfect, perfect. Over to you.
Right. Okay. Hello, everybody. I sincerely thank the organizers, for this wonderful opportunity. I will be presenting NeuroSAFE Tech story now. It is the name of the solution developed by Transcell, leveraging its credibility in human stem cell technology, complemented with the next-gen digital assets and robotic process automation-like framework. Transcell as a group enjoys working relationships with a few well-known academic and IT giants of global repute. Many countries require their citizens to receive, certain immunizations, and even there are laws that dictate mandatory vaccinations, such as those required for children and now for COVID-19. Controversies over the safety, efficacy, and morality do carry historical baggages. For example, vaccine neurovirulence like safety concerns with terminologies like paralysis, Guillain-Barré syndrome, progressive nerve disorder, associated with the, some of the vaccines like polio, swine flu, hepatitis B, which have been in the global immunization programs for long, declared as adverse event in the immunized population. Then in our lifetime, we have experienced the neurological debilitation caused by well-known human neurotropic virus, which is, SARS-CoV-2. We have real-time evidence of Guillain-Barré syndrome caused by J&J vaccine, in addition to FDA black box warnings given to Moderna vaccine and Pfizer-BioNTech for their mRNA vaccine types as well, alluring to the point that it is not just the inactivated or attenuated vaccine types that are at the risk of causing neurovirulence, but any vaccine that is structurally or functionally mimicking either the neurotropic virus or viral particle is at the risk of causing this not so good adverse event in the immunized population. To that effect, in fact, WHO mandates testing neurovirulence for polio, mumps, measles, rubella-like vaccines, and I'm sure COVID-19 and monkeypox vaccines are added to this list. It is our belief that it's like a moral obligation as well to periodically assess the seed stock for any neurovirulence risk, in addition to the preclinical stage requirement for regulatory submissions. So MNVT, which stands as monkey neurovirulence safety test, is the gold standard method known to the industry to test, while non-primate model-based exploratory work can as well be found in the literature as some alternatives or complementary methodologies that are work in progress. With the MNVT as the gold standard method practiced, COVID-19 pandemic has triggered the issues on ban on monkey exports from China, breeding monkeys, leaving the industry and global scientific community high and dry. Then adding fuel to that shortage of monkeys, US House has passed legislation to end FDA animal testing mandate in June 2020, which is this year. And in 2020, FDA, CDER like agencies published certain high impact factor review articles encouraging the global industry to adopt new approach methodologies that yield human mechanistic readouts. So as you see, there appears some deja vu or compelling situations to look for non-animal new approach methodologies to adopt in the workflow. And also, you all will be very pleased to know that last year, Merck, which is a very highly respected group in this domain, endorsed the concept of human-induced pluripotent stem cell technology to screen vaccine neurovirulence based on peer-reviewed publications supporting the very concept itself. Now, allow me to introduce Transcell Oncologics, dedicated vertical Transcell Bio that has all new league of human surrogate in vitro microphysiological systems. The vision of this, dedicated vertical is to offer safety efficacy testing as a solution to the global pharma and biopharma industry, while the mission is to offer non-animal cruelty-free solutions. Few of the residing human surrogate in vitro systems like trans-MSC are human pluripotent stem cell configurations dictating the very use case itself. So some of the attributes like phenotypically responsive, genotypically reactive, and functionally approachable are special to Transcell Bio residing platforms. And at the same time, the source that we have identified to be relying upon is human biological discards to harvest these platforms and then configure in our labs. So we believe that as long as the human race exists, there would not be any dearth of this raw material, that is very crucial in harvesting and then producing these platforms in volumes that becomes the basis for some of these alternatives, methods and methodologies and systems that we have developed to the existing approaches and methodologies. So NeuroSAFE is our product that is offered as a solution with the two components. The first component is this TransMSC, which is human-induced pluripotent stem cell configured in vitro platform. And the second one is trained software that is embedded with artificial intelligence and machine learning tools that can predict signals from test material treated TransMSC. So in essence, it is a revolutionary product offering that can be seamlessly integrated in the user's workflow. Yeah, to implement this solution, the user actually requires no additional infrastructure added to their facility also. And also, I'm very proud to share with you that, some of the specifications of this offering are even reviewed by agencies like ICCVAM, which is a permanent committee, incorporated by NIH. To continue talking about NeuroSAFE, I also want to share some very out-of-the-box advantages with the adoption of this technology in your workflow. Like, once adopted, the user can utilize the assay system in preclinical stage or clinical trial stage, or even real-time manufacturing stage. And the second one is it is rapid. The assay system is rapid and made robust with increase in the volumes of data being ingested into the software. The third interesting feature is it definitely reduces the overall cycle time in testing or predicting whatever, either it could be safety or the associated concerns. That is part of the test material. So in fact, it is very significant at the same time being specific to human species with absolutely no extrapolation needed of the data, that is generated or routed through NeuroSAFE. Then the next point is the assay system can very well be adopted for routine testing even before releasing the batches. The next again is the same framework is tested to be suitable, in fact, to get a dedicated module in detecting microbial contaminants, adventitious agents in the product. And then last but not the least, a feature is that, it can become part of the process to detect neurovirulence signals even in pharmacovigilance. So overall, we believe that it is only to do with benefits and advantages and absolutely no risks of any kind. If a user decides to adopt NeuroSAFE in their workflow or process, and ultimately, it is kind of implemented specific to the product and a process that is followed in the user's operations. So in essence, NeuroSAFE supports in adopting cruelty-free operations, and most importantly, the feature of which that is so quintessential to adopt for routine testing is one of the stark advantages we believe that is offered to the user. We have protected, in due course of our development and implementation in the early adopters facility, we have protected the details of the solution offering as our intellectual property, validating its industrial application. So, in a nutshell, if I have to explain the scheme in implementing NeuroSAFE in your facility or workflow, like you have your test material, which can be an aliquot of, the batch that is produced. And this test material will be advised to be incubated with trans-MSC units that will be supplied, to the user, and the user can be anywhere in the world because the logistics of shipping these trans-MSC units from India is very well established in our hands. And this incubation period can be between 3 to 4 hours. And after the incubation period, the phase contrast, inverted phase contrast microscopic images, again captured with some specifications followed, will be fed into the trained software, the access of which again will be given to you through cloud infrastructure. So, practically, there is nothing additional that, you will be expected to have in your operating process, other than what we supply as trans-MSC units, and then the access of the software is also given to you. So after feeding these phenotype images into the software, within 5 to 7 minutes, the score will be published in the form of reports. So the score could be, the cellular phenotype based neurovirulence or neurotoxicity, and depending on what are the options, the reports also could be pertinent to the sterility, of the test material in discussion. And as I said, the extrapolated advantage of understanding, the neurovirulence signals as adverse events in pharmacovigilance as well is part of this entire offering. The software component follows certain business rules, while sourcing data followed through annotations till analyzing to publish the same. So this is kind of a robotic process automation that we actually have followed while developing this solution, and at the same time, when it is implemented at the user site also, this is a continuous kind of handling, the data and then training the machine. So machine learning and then machine training is made continuous in the NeuroSAFE assay system, again, owing to one special key that is part of this entire build-up. So this special key is, has the ability to continuously ingest and annotate the data while this solution is implemented at the user site. So it makes it a dynamic process, and then the volumes of data being ingested directly kind of influences the robustness of the assay system in predicting the readouts. In the NeuroSAFE context, it is to do with neurovirulence or neurotoxicity, and then even the sterility aspect of the test material. So it's a totally next generation, as we believe, in its offering as a workstation solution. Definitely, to understand your assets better and faster, as we have, made it so comprehensive by combining, the human in vitro surrogate microphysiological platforms with image and data analytics seamlessly integrated in the workflow. I thought we'll show you a representative NeuroSAFE routed phenotype data set, which is first of its kind, and then we believe that these are some signals that we want to kind of show to you, and you wouldn't find any of these images in the literature so far. I mean, unless we publish and then we kind of decide to communicate both the phenotype and then genotype data sets routed through NeuroSAFE on human-induced pluripotent stem cell platform. So this particular signal, speaks on the test materials in neuroinvasiveness, neurotropism, and neurovirulence to the extent of quantifying the signals as well, but captured from the microscopic images yielding the signals. And also, we took clues from WHO set neurovirulence scoring method in quantifying affected cells in building the prediction model on foolproof Amazon Web Series specific cloud infrastructure. So, in fact, we actually went to the extent of even classifying these affected cells as, cells in shock, apoptotic cells, and necrotic cells. So the prediction model is trained in such a way that it can detect the signals that are heterogeneous and continuously, the machine is trained with those signals. And again, if any assay system is built on platforms, I mean, the components and the platforms as well, should be peer reviewed and regulated. So since we chose AWS as the platform for, launching the regulated digital framework, it is in fact assured in terms of the quality of the readouts that are part of this assay system. And certain advanced algorithms like neural networks became the foundation of NeuroSAFE's digital framework, again, embedded with the artificial intelligence and machine learning tools. Likewise, as I said, the components have to be both peer reviewed and regulated. The first component, which is the human-induced, pluripotent stem cell tech platform, we take pride in sharing with you that it is very well characterized when it comes to the kind of gene markers and then also the proteomics, that are expected to be expressed on this platform, including the population doubling time establishment, as well as the stability and genomic integrity of the very primary and progenitor cell-based platform that is part of this NeuroSAFE offering. Now, everything bogs down to NeuroSAFE's application relevance as a non-animal alternative strategy to reduce, replace, and refine animal use across the stages of vaccine production, echoing the 3 R's in animal use. So since I think 2011 onwards, globally, some of these known agencies that have been encouraging the industry to look and adopt for non-animal methods and strategies to test both human vaccine potency as well as safety related concerns. It's actually very encouraging to know that not just the suggestions, but also in detail, you would get to read the rule books to look for some accreditations, as well as to follow the alternatives and then strategies to be part of your process and workflow. To summarize, NeuroSAFE is kind of being introduced to you as a must and good solution to have in your system or workflow, and it is demonstrated to be applicable to vaccine candidates that are in development. Again, when I say vaccines or candidates, I'm only referring to the vaccines that are developed for neurotropic pathogens. So neurotropic pathogens could be both like, virus or bacteria. And NeuroSAFE is applicable to even licensed vaccines because we have some experience working and supporting some of the companies that have the vaccines already in global immunization programs, but they want to kind of explore routing through non-animal new approach methodologies for their batch testings. And then the third item is like NeuroSAFE is applicable to vaccines in immunization programs and for you to continue to participate in safe vaccinations. And the last one is like, you definitely have to be cognizant of those vaccines that have some, dubious episodes of virus inactivation. So for example, rabies or mumps and also polio. So there is always this compelling need, although, these vaccines have been in the global immunization programs for 50 plus years, there is still, scope for manufacturers to adopt for NeuroSAFE-like testing strategies to generate, to continue to generate human-related signals. So when we offer the NeuroSAFE as an implementation program in the user's workflow, we go stepwise. So step one is definitely like, to establish the very relevance itself. And then step two is after establishing the relevance, it all depends on like, the product that is chosen by the user. So specific to the product, the range of pathological patterns on trans MSC will be established as standard curve. And then these data points would be used in training the NeuroSAFE software. So these product-specific data points would become second level of benchmark panels in the user's scheme, and ultimately supporting the use of the assay system in routine, batch-specific QC testing. So I just want to make a statement here that, at any given point of time, human data definitely like, takes precedence to any kind of or any animal data that, for ages, the industry has been generating and believing that, that is good even in the context of human species. So thank you so much.
Dr. Subha, thank you so much for that talk now. I think it was an absolutely amazing talk, and it's really, I think, impressive to see the advances that you have made in the field, or you and your group. I think we're probably going to open up the floor now for questions. Melissa, I think, if I'm not mistaken, people will be able to either raise their hands or to post questions in the Q&A, and then we'll run you through them one at a time, both Shri and Subha. So, Subha, maybe just to open up the questions while we wait for the audience to post some of the questions. Just a quick one from me, I think, I think the crowd on the call and the audience in general, when it comes to AVMI, really come from a background of vaccines and biologicals, and they really are manufacturers. So with respect to this product, it's a new product. It's super exciting. It seems to like it's planning to be a game changer, especially in terms of bringing products onto the market that negates the need for animal workups. So my question really is that, the kit as it stands now today, the NeuroSAFE assembly, is this a fully GMP approved product at the moment? So in other words, if there's any accreditation linked to your product, can you share that with us? And also maybe in which domain? So for example, FDA, EMA, etc. Dr. Subha, are you still with us? I think... Shri, are you still with us? I think we've lost Subha.
I'm with you.
Is that
Yeah,
perhaps a question that we can extend to you?
Yeah, absolutely. I can answer that. When it comes to the GMP practices, we would say it is an innovation, and it definitely follows all the GMP practices. When it comes to collaborator, right, just example, the collaboration with the vaccine manufacturer and getting it to the regulator, that's what your question is. I mean, if you have any regulator-specific data, that's what you're saying, right? Am I correct in my understanding?
Yeah, no, 100%, yeah. Dr. Shri, for some reason, we have a bad connection with you. I think it's very static, but we don't want to lose you while we're waiting for Dr. Subha perhaps to return. But there was a follow-up question just from one of the audience members in terms of whether NeuroSAFE at the moment has been used on any approved licensed vaccines in terms of releasing it into the market.
Okay, right now it is being used by firms who are actually using it for vaccines which are under development and which is due for launch. That's number one. Number two, it is also used by companies which are manufacturing anti-venom vaccines. That is, there are two use cases here. One is where companies see this as a value for research and development, under launch, very strict protocols to follow. And there is a new vaccine in the market, so they want to ensure that it is a hit in the market. So they are using NeuroSAFE because number one, it solves the animal cruelty part. You're not using animals. At the same time, it solves the safety and potency. It also brings down the vaccine manufacturing time from 28 days to a few hours. So that is the key, if you ask me, because vaccine manufacturing per se, you know very well that it takes a long time to manufacture and ensure the safety and potency of it. So this solves all those key problems in the industry.
Yeah, 100%. Dr. Subha, did I see you back now? Did you join us again?
Yes, please. My apologies. I don't know. I was logged out automatically. Yeah.
Thanks for rejoining. Don't worry, your colleague Shri, he's done a wonderful job just in covering you in your absence.
Thank you, Shri.
Just quickly though, quite a few questions we've run through Shri already. But just towards the end of your presentation, I think you made a very interesting comment just with respect to the progression of any candidate vaccine or therapeutic through the early stage development into more your late stage development. And quite often our products have to migrate from animal models then into people, right? And we know it's a very tricky transition with quite a few of the vaccine candidates, at least on the market today. You don't necessarily get very good readouts on vaccine safety. And it's definitely not necessarily a very good correlation back to what's going to happen when you go into humans. So my understanding here with respect to your stem cell technology approach is that your cell lines would almost certainly be derived from humans, I would assume.
Yes.
Is that correct, first of all? And then secondly, if that's the case, how easy is it to get, regulatory approval for the use of those stem cells? And how do you make a decision in terms of phenotypic and genotypical advancement in terms of which cell lines to go for?
Very good question. Yes, for the first question on your assumption that these are human derived, stem cell configurations, and that is right. And the second question, how do we, make sure that it is a regulated practice, right? In terms of using. Yeah. So, Dr. Ebrahim, you will be very pleased to know that we thought about it, and we very well kind of knew that it definitely, the source has to be ethically, accepted, either ethically accepted or ethically immune, right? And then at the same time, there should not be any dearth of the raw material that we have chosen, because if something needs to be developed as a product, it has to be produced in large volumes, and then reproducibility, repeated use of the platform, essentially in the assay system is a must. So you will be happy to know that the source, the raw material here for us is human biological discard, which is human umbilical cord tissue. So as long as the human race exists, we would not have any dearth of this raw material. And at the same time, it's an ethically immune material, because we are not causing cruelty to even the donors. It's because it's a biological discard. If we do not collect it, it would go into garbage. It would go into the dustbin, right? So we have taken care of the ethics aspect of it, and we have taken care of the continuous supply of the material. And we actually have worked on this particular source for the past, I think, 10 plus years, and have very well characterized in terms of the signatures, basically, and those signatures happen to be, of course, 3 types. One is the phenotype, the morphological variations, and then the second type is genotype expressions, and the last one is proteomics, right? So we have correlated the morphological perturbations with genomic expressions, and also have meaningful relationships established with the protein expressions also. So it's like a dynamic process, barcoding each of these signatures. That is part of the background work that we have undertaken before bringing this particular platform into the assay system, NeuroSAFE, that we have developed.
Yeah. Excellent answer. Thanks so much for that, Subha. Just as a follow-up to that, I can only imagine, and again, we have no experience on this within AVMI, so that's why we need experts like yourselves to guide us through this process. But in terms of approval through different regulatory authorities, what is Transcell's approach to this? So you're probably going to have to seek approval from, let's call it, the FDA and EMA. And I guess the more your product gets tested in preclinical setups, the more data you'll gather that will validate to the regulators that this is, a safe and also a feasible way of evaluating neurotoxicity in candidate vaccines or therapeutics. So just with respect to your approach with the regulators, how do you foresee this rolling out?
Right. Good question again. In fact, we have been following, a 2-pronged strategy. So in our NeuroSAFE's universe, we have actually started implementing this in early adopters workflow. So we believe that the data that is generated in their workflow, the early adopters workflow, is very important to go to the regulators. But we believe that the early adopter is empowered to go submit the data to the regulator and get the necessary approvals, to utilize this in their workflow. That is one approach. And the second approach is we are kind of wanting to make NeuroSAFE visible and its use case, and then the application as, non-animal and new approach methodology that FDA itself has coined this term, but we sincerely think that, the adopter should, or can take the data to the local regulator. We have actually submitted some early stage data to even US Pharmacopoeia, and then ICCVAM is another agency that we have had interaction with. So it is the data that the regulator would want to see, and not the methodology. So so this has been our experience, and then we think that is the way to go, especially for non-animal methods where, and that too for this industry, vaccine industry, where day in and day out, you kind of experience the context and scenarios where, cruelty is caused to animals.
Yeah. Thanks so much, Dr. Subha. Again, just a reminder to the audience, Dr. Subha did finish a presentation earlier, so we can keep her. We've got until 3 o'clock. We don't have to, but Melissa has just given you all permission. So again, if you want to ask questions, just raise your hands, and I think you'll be able to unmute your mic as to follow up questions. But while you're thinking about some questions, Subha, I've got you now, yes, so I'm going to hold on to you for a few minutes. Just quickly, with respect to, I was very interested in the talk just from a cost of goods point of view, because obviously I think one benefit to NeuroSAFE is the idea that you would not need to go into animals, which we know from a ethical point of view, is just a massive advantage. And I think all the funders are asking more and more so that these approaches get taken. However, though, I would be very interested to understand what sort of savings you foresee companies or manufacturers potentially benefiting by not going into animals, which are quite expensive, and being able to get away with these types of in vitro cell-based lab methods. Have you done any workup to that sort of evaluation? And can you share that with us as the audience? Yeah.
Yes, please. See, if somebody is into routinely, going to, or have to be doing the animal testings, of course, the vaccine company is expected to, even post-licensing, and then at every stage, periodically, these have to be tested. So if it is animal testing, you have to have the animal facility, or you have to send your material out to a centralized facility, which is routinely doing the animal testings. So that is a big expense, and we very well know that, not anybody who is not serious, can't afford to just step into it. And then the other one is, yes, if you need to maintain the animal house, you also have to get into the supply, import, or supply of these models also, right? Animal models also. So breeding is one, and then taking care of the resources, the technicians. We understand, and then we have had few interactions also with the workforce. They are so demotivated, to be doing day in and day out what they're expected to be part of that job profile. So these are some of the bane, disadvantages, not just with the cost, the cost or pricing aspect of it, but also with the moral of the very team and then the group. Yeah. But if somebody is part of this NeuroSAFE adoption, it's such a good feeling that, you're actually part of, you have this opportunity to be part of this high-tech access system, and then you're part of this digital, transformation that your organization can go through. And then what is your expense? It is only very kind of meager in terms of accessing this software license, and then procuring the trans-MSc units. So it's very, very affordable, and then economical, if I have to compare these two assay systems.
Yeah, I would have thought the same. Just off the top of my head, animal models cost a lot of money, so I would have assumed exactly the same. Dr. Subha, thanks for that answer. We've got Matthew with his hands up. Matthew, you're only allowed to ask a question if you switch your camera on. So Melissa, if you can unmute himself. Matthew, over to you.
She's not allowing me also to switch on the video.
Matthew, unfortunately, there's a lot of echoing on your side. Not too sure why. Maybe I'll just mute.
You're muted, Doctor.
Is it echoing
well said. Okay, brilliant. No, thank you so much for that. I just wanted to know, was the actual NeuroSAFE of time frame from, let's say, starting the test and getting your end result?
Maybe Matthew can type. Dr. Ebrahim, you also have to unmute.
They can hear me now.
Yes, I heard somebody. Dr. Ebrahim, you may have to unmute, please.
Oh,
yes.
Sorry about that. Matthew, we couldn't hear you early on. If you can just maybe try again.
Can you hear me now?
Loud
Yes.
and clear. Loud and
Okay,
clear.
sorry about that. I just want to say thank you so much for your time and the great presentation. I know with the typical, the MVNT test, it takes quite a lot of time to get the release result. I'm not sure if I may have missed it in your presentation. What's sort of the turnaround time from the result getting released from the NeuroSAFE equipment?
Matthew, kindly repeat the question. There was a pause in between for me. Yeah.
Okay, so I think I'm just going to type the question in the box. I think I've just got a bad signal here. I'm sorry about that.
Yeah, I think, Subha, if I can maybe jump in there, I think what Matthew was trying to ask is if when you do a test, for example, a test run with your NeuroSAFE kit, from the starting point to when you actually end up getting a result, in other words, the workup of the data, how long does that typically take? Animal models and animal studies, we know it's weeks, can sometimes even take months.
Yeah.
So just in comparison, what sort of time frame are we looking at?
Well, 24 hours.
It's too fast for you, Matthew. You'll be out of work.
That's brilliant. Sorry, man. I think that's just really great. And I mean, I know that with such a good turnaround time of 24 hours, I'm pretty certain in the future that can maybe even get shorter. But when clinical trials are done, sometimes when the vaccine doses are made, I guess sometimes there might be some variability or incorrect dose. So I mean, was there any envisionment to make use of this, for clinical trials to check whether those doses are still fine to use?
Of course.
Part of that, yeah, and I mean also, I was thinking that because it's such a small device, I'm not too sure what's the QC requirements, for running that, but whether even, I'm just thinking on the top of my head whether nurses or whoever administers that, they could use it as a early in the morning before they start vaccinating people. It's quite a nice piece of equipment with that short turnaround time. I guess that's still a far thing away, not really going to happen, but more for the clinical trials, so you can save a bit more money there.
Absolutely, I think it's a very good point, and then I mean, it's very much applicable, I would say.
Yeah, thanks so much.
Yeah. Thank you so much for the question, Matthew. Subha, we are running out of time, but I can't stop myself. I've got one or two more questions. Just a quick one though. So obviously, when you're investing in any piece of equipment, any piece of hardware, you're investing quite a bit in terms of software as well. You'd obviously want that, that piece of equipment and that software to serve you across multiple purposes. So probably a tough one for you and for Shri, but I invite Shri also to comment if you would like to. Just in terms of your business development strategy, you started off now with NeuroSAFE, and obviously it's a lot of investment. You guys are trying to find your feet in the industry as well. But what is your follow-up plan? So after being able to hopefully successfully roll this out and having a key marker in terms of neurotoxicity in the field, do you plan to utilize a very similar or even exactly the same platform maybe to test for other indicators as well?
Brilliant question it is. Yes, we do have the other extended, workstation solutions like CardioSight, which is to test cardiotoxicity, which is equivalent to, again, CIP, a comprehensive in vitro proarrhythmia assay. That's one in our arsenal. And the second one is hepatotoxicity, to assess the drug-induced liver injury also. That is another workstation solution that we have with us. And the third one is hemotoxicity, to test the blood toxicity related concerns of the test material. So it's not the same, I mean, NeuroSAFE platform extended, but then like similar kind of a strategy that we have utilized, while the stem cell component is the one that would vary for the kind of readouts that are relevant to the particular assay system. For example, if it is hemotox that I'm referring to as a workstation solution, it's not trans-MSC, which is the component one there. It is trans-HSC. Trans-HSC is, it mimics the human blood system, in vitro, human blood microphysiological system in vitro. So that is the component for that particular assay system. So like that, we have close to some 42 plus assay systems, developed or being developed as workstation solutions. Yeah. So it's kind of quite a battery of testings, that we envision to be part of, offering, as new approach methodologies to the global industry. Yeah.
Wow, sounds amazing, Subha. I can only wish you all the success, but I think you've got grand plans and it's very exciting to hear about them. Just a final one from me, and then unfortunately we have to close the meeting. Just logistical support globally. Do you have any procurement networks that anybody interested can go through? And also, in terms of distribution and getting products into Africa, do you have any plans in terms of expansion and getting the logistics worked out for African manufacturers, for example?
Brilliant. Yes, I mean, we are quite open, and then we are exploring, to engage discussions with the potentially interested groups who would become our partners in Africa. Dr. Ebrahim, we are quite open, because the assay system, as you have understood it right, it works in such a way that logistically it's very kind of established. You were even to supply trans-MSC units, and then to give access to the software, it requires, I mean, there are no systems or hurdles in between. Yeah, so that is a possibility in our plan, to enter into Africa.
Excellent. Thank you so much. Sorry, someone commented. Oh, Shri, go for it. Yeah, please.
I can just add to that, to what Dr. Madam said. I would say BioVac and AVMI, right? Mr. Patrick and, BioVac has been very, very, kind to, hear our story and, listen to these innovations per se. So I think BioVac as a partner is an absolute win-win for both of us, and we would like to definitely work with BioVac both for vaccines which are under development and which are already in the market, and use AVMI's help to spread that to all the countries throughout Africa, both south and north. So that would mean that we both are winning the game. Yeah, 100%. And Shri, thanks so much for that comment and that invitation. I think, in closing the meeting off, just quickly, I mean, we had the broader AVMI audience here today, so hopefully there was participation from all over Africa. We invite any particular interested party to either reach out to you and to Subha directly. And if not, you feel free to contact me or Melissa, and we will absolutely put you in contact with the group so that you can expand those talks further. I think for sure, BioVac will probably have some sort of an interest. And so into the foreseeable future, and this was the first stepping stone towards just advancing your technology across the African continent. But we would encourage those further conversations with BioVac to happen in a, in an offline sort of one-on-one fashion. So hopefully over the next couple of weeks, we will definitely do so.
I just would like to take the final few minutes just to thank both you and Subha. First of all, Subha, for taking the time and also for taking us through the presentation. I think it was an excellent presentation, quite simple and easy to follow. And Shri, of course, thank you for your support as well. Just before we close, I would like to also extend just a warm thanks to the AVMI audience for taking time out of your schedules to be able to take the call today. So thank you, everybody.
Thank you, Dr. Ebrahim. Thank you, Melissa. Lovely. You know, it has been such a great experience interacting, you know, in this session. Yeah. Thank you.
Excellent.
You're welcome. Thank you, everyone.
Take care, everybody. Have a good day further.
Bye-bye.
Bye. Bye-bye.